So good evening. Thank you for inviting me to talk today at this really fantastic workshop. I'm Josh. I'm a postdoc at the Pirbright Institute in the group of Professor Munir Iqbal. The group is mostly concerned with characterizing avian influenza viruses and developing veterinary vaccines. So today I'll talk about interactions between receptor-binding, immune evasion, and glycan shielding. So just a quick intro to my subtype of choice. I've been working on the hyperendemic poultry-adapted H9N2 avian influenza viruses. These are typically low-path avian influenza viruses that are present in a vast geographical range. In SPF conditions, they typically present with reduced morbidity and mortality, but often in the field, it's a very different case, and you get co-infection with other avian viruses or bacteria, and that leads to enhanced poultry mortality and morbidity. I mean, a lot of my work is on H9N2s, but I think some of what is learned or can be learned can be sort of extrapolated to other types. On the topic of mechanisms of antigenic drift, I've got a quote from a paper by Jonathan Udell here, where a key feature of immune escape may be modulation of HA receptor avidity, which triggers epistatic alterations, secondarily modulating antigenicity. So this sort of statement encompasses the two main mechanisms of antigenic drift that I'm going to be talking about. So here is the influenza HA protein that we all know and love, a very simplified version with these simplified mechanisms of antigenic drift. So one mechanism, or first mechanism, involves mutation in the head region of the HA protein that increases or modulates binding avidity, so that the HA can outcompete antibodies and bind to sialic acid. Second mechanism involves the introduction of mutation that introduces a glycosylation motif, and a glycan at these sites can allow shielding of epitopes to prevent antibody binding. A third mechanism is where a distal mutation to the head limits sort of the mobility or flexibility of the head region that limits epitope accessibility. And a fourth mechanism is where a mutation occurs that directly changes the epitope's shape or structure of the HA protein. So work that was previously done by Tom and also by Will looked at work where they looked at the genetic basis for antigenicity in H9N2 viruses, and this is work that really involved lots of HI assay data using panels of mutants with chicken antisera and monoclonal antibodies, and production of a model, and then predicting mutations and testing those to see how accurate the model is. But what this essentially showed was that there were 7 mutations that significantly impacted antigenicity. 4 of those introduced an endemic glycan, and 3 regulated receptor binding avidity. So this really sort of underlines the importance of those 2 mechanisms to antigenic drift, potentially. Follow-on work from that looked at the other side of the coin, the sort of genetic basis of receptor binding, really. So again, in H9N2 viruses, assaying by biolayer interferometry these mutants and forming models. And what this showed really was that, or identified, was some of those mutations that could be identified from this work, that mutations that regulate binding phenotype also sort of correlate back with impact on antigenicity. And you could also somewhat predict binding phenotype by the impact that a mutation would have. So if it would increase the net charge of the HA protein, then it may be quite likely to increase binding avidity for receptor analogs. So in some work that I did, where it characterized antigenic diversity in contemporary H9N2 viruses, which involved isolating viruses from Pakistan and generating a panel of reverse genetic viruses that represented the contemporary population, and screening these with chicken antisera to look at the impact of basically to identify antigenic diversity. And what this found was that mutations at residue 180 had a up to 4 log change in HI titer. So it's important to say that at this stage, this is chicken antisera that was raised against a virus that has an alanine at residue 180. So those mutations which naturally have a T or a T or a V had a significant HI titer reduction. We then went on to show by bilayer interferometry that these mutations impact binding phenotype. So these, typically of H9N2 viruses, they preferentially bound to the 6-sulfated 3-acetylene receptor analog, which we use. There was capacity in the high avidity T180 virus to bind to the human-like 6-acetylene in blue there. Analog, and when you mutate this, so when you change the T to an A, you get a quite a significant reduction in binding avidity, and you also lose the human-like binding capacity. Likewise, if you go from a T to a V, you significantly or you further increase the binding avidity. So these viruses bound to all tested receptor analogs even stronger than going from an A to a T. We looked at the impact of these mutations on viral fitness in vitro in MDCK mammalian or CKC avian cell lines, and we showed that these are called adsorptive mutants when they have these avidity enhancing mutations. So these adsorptive mutants were attenuated in cell culture at least. So at various time points, viruses with an alanine grew better than those with the T or V at residue 180. So this brought a lot of previous work, for example, by Scott Hensley and Jonathan Udall, have looked at modeling antigenicity through mice or various systems, looking at escape mutations and also looking at epistatic mutations. So if I just talk about, should I get a pointer? Just this first. I'm calling this a fitness landscape. So this sort of black line as it drops, the fitness of the virus drops. So what they did is they showed that passage of a virus through a vaccinated mouse led to an escape mutation, but that mutation came at a cost. Passage of that mutant through a naive mouse led to an epistatic change. And when looking at the actual impact of those mutations, the escape mutation increased binding avidity and permissive mutation reduced binding avidity. So in a similar light, I wanted to look at how this works, because we had these viruses which were attenuated but antigenically distinct for H9N2 subtype, and so I wanted to look at mechanisms of epistatic change. Epistatic mutation. So I looked at N-link glycosylation. It's also involved in antigenicity, so it would be an interesting place to start. And so from looking at the literature and looking at other sites that often glycosylated or have an impact, I chose these 3 glycan mutations, or 2. One sort of cropped up by itself after passage in egg. But so yeah, I made a panel of viruses either with A, T, or V at 180, so various different avidities, and an introduction of glycans 134, 148, or 189 in the H-shape head domain. And the bilayer interferometry showed that addition of these glycans does modulate avidity in the various backgrounds. So they generally have an impact where they reduce binding avidity to tested analogs, but definitely a position-dependent effect. So a glycan at residue 148 had quite a more significant impact on binding avidity. So in that virus, say with V180, addition of a 148 glycan in abrogated human-like binding and significantly reduced avian binding. Yeah. So then we wanted to test, see how these viruses did in cell culture, and again in the two mammalian and avian cell lines, and we showed that that yes, indeed, these glycans, when added to a higher avidity background, they do improve viral fitness. So for the T180 and B180 viruses, addition of these glycans, and particularly 148, led to improved replication kinetics at various time points. On the other side, when you introduce these glycans to a relatively low avidity virus with an alanine registry 180, you attenuate virus. So I guess you can attenuate virus either by regulating avidity or introducing an alanine glycan, and possibly epistatic mutations can go either way. So lastly, we looked at the effect of these absorptive or avidity enhancing mutations and alanine glycans on antigenicity, both in isolation or together. So this first panel over here is just sort of an extension of the very first HI assays, but basically using sera raised against a relatively low avidity virus that has an alanine. A lot of these mutations, whether they introduce an avidity enhancing mutation or whether they introduce a glycan, led to up to 4-fold reduction in HI titer. When using sera raised against the high avidity T180 virus, you can sort of ameliorate a lot of the antigenic effect, with the exception of a virus that carries the 148 glycan in the T180 background. You still get a significant amount of antigenic diversity. So this sort of indicates that maybe a path to a fit virus that has maybe initially escaped gain of permissive mutation and is still antigenic divergent. So in summary, absorptive mutations facilitate immune escape, attenuate virus, compensating mutations through ending glycosylation restore viral fitness, and alone and in combination with absorptive mutations, ending glycans can exert antigenic diversity. So thank you very much for listening. This is just the group at Pirbright, and this is Munir, who I think was supposed to be here. Maybe he's not at the moment. Thank you.